Hi, everybody, and welcome to Echo Live. My name is Anna, and I am coming to you from the Michigan Science Center's Echo Distance Learning Program. Today, I'm not revealing the title of our episode just yet because it is one of my favorite days. It is, again, Dissection Day here on Echo Live. Now, I'd like you to guess what we might be dissecting today, and this is a good chance for you to practice using the chat or the comments feature of whichever program you are joining us through. If you are in YouTube Live or Facebook Live, you can take a guess as to what we might be dissecting for today's Dissection Day by telling us in the comments. If you are joining us through Zoom, like Elise is, hello Elise, tell us what you think we might be dissecting. Last week, we dissected a sheep eye to talk about how humans perceive vision. So how vision actually works inside a mammalian eye. So that was a dissection of just one organ. Now, today we'll be dissecting an entire organism. So maybe that's a clue to some of you. So instead of just one part of an organism, such as an eye, today we'll be dissecting an entire organism. Does anyone have a guess as to what we might be dissecting? Jim thinks maybe a lamb. Jamie also agrees a lamb. Interesting guess. Couple guesses in Zoom for possibly a frog or maybe a mouse. Elizabeth thinks a cow. Now, we said we'd be dissecting an entire organism today, and I don't quite have the space in my studio to be dissecting a cow. So nothing quite so large. Think a little bit smaller. Think pretty accessible. Now, this is a dissection that you can actually pretty easily try at home. Maybe that gives you another guess. So think about your guesses. Think about the organisms that you are guessing. Remember that this is an entire organism, not just one organ. So think about that. Oh, a couple great guesses. Today we are going to be dissecting a flower, which is an organism. So a lot of guesses for animals, but not all organisms or living things are animals, right? That's just one kingdom of living creatures. Today we'll be dissecting a flower. So I have some different flowers here inside my studio, along with my dissection tools here on the tray. And this is a really easy, very fun dissection that you can do at home because you probably have flowers growing somewhere near where you live, or you could pretty easily get them from the grocery store, or maybe even one of your neighbors. If you aren't growing flowers in your garden right now, one of your neighbors might be willing to donate one to you, but always make sure that you ask first. I actually got most of my flowers here in the studio from one of my neighbors. So they donated these to science so that we could do a flower dissection today. So I've got a good variety of flowers here. I still have our carnations, which have definitely seen some better days. These are the carnations that we dyed on Earth Day. We also smashed some of these yesterday on our liquid nitrogen episode of Echo Live. We might not dissect those because they're not doing so well, but we've got some other options here. We've got some daffodils. I have a peony, which is this big, really, really pretty flower that grows on almost a tree or like a bush instead of just straight out of the ground. I've got one last daisy left over from our Earth Day episode and a couple of tulips as well. So we've got a variety of flowers here, and they all look very, very different. Now, just by looking at the flowers, what are some differences between these flowers? Think about what they look like. Think about their different features, because they certainly all look very different. So what is one way or one difference that you could find between these flowers? A way that we might be able to sort them or classify them, or just something different you notice between the flowers? Go ahead and tell me in the chat or the comments feature. Ooh, one great answer, it came through in Zoom, that says the smell. Someone tells me that there's probably a different smell to each of these flowers, and that is completely true. Now, you might not be able to tell what they smell like from here. I can definitely smell the flowers, and I can also tell you that my allergies are going a bit crazy. Since I picked these flowers, they're starting to lose some of their pollen or release it into the air, and so they smell very beautiful, but it's also making me sneeze a little bit. So if I do sneeze throughout the program, bear with me. We'll get through this together. It is pretty pollen-y here. But other great answers are the size. So some flowers are pretty small, whereas others are fairly large, especially our peony is really, really big. Some people said the petal shape. Yeah, so some of them are quite round, like on our tulip, whereas others are maybe a bit more triangular, like here on our daffodil. The color is certainly different. So we've got yellow, pink, white. We even have these rainbow colored carnations that we dyed using food coloring. So those aren't necessarily their natural colors, but they are still hanging in there. And some people are pretty impressed I've kept them alive this long. And let me tell you, so am I. Great answers, everybody. So there are certainly differences between these flowers. But now let's think about why. So these flowers certainly look different. They're different colors, different sizes, different shapes, different heights. They smell different. Can anyone imagine a reason why it might be advantageous or a good thing that flowers either look or smell different? Why might that be the case? Why might flowers look or smell different from each other? So think about all the things you know about flowers. Think about the flowers that you either have growing outside. Are they the same or different than ones that we find at the store? Why might that be the case? Elise says that maybe they have different purposes. Jason tells me that maybe it benefits different pollinators. Those are great answers, everybody. So that is exactly the answer we're looking for, is that these flowers look different because sometimes they naturally grow in different environments, but also because they are designed to attract different types of pollinators. So last question for the group before we get started here, because it's an important one. Could you name me a type of pollinator? So we said these flowers look different because they are attracting different pollinators. Pollinators are things that help to spread the pollen between different flowers so that they are able to reproduce. Does anyone know a type of pollinator? Lots of great answers starting to come in. Tell us, there are hundreds of pollinators, thousands even. Think about bees. Yeah, some people telling us bees. Lots of guesses for butterflies. A couple of our friends in Zoom tell us all sorts of flying things like bees, moths, butterflies, even bats or hummingbirds. You guys are experts on pollinators. Pollinators are super important, and they're not just the common ones that we might think of. Pollinators can be those flying organisms like we talked about, bees, butterflies, wasps, moths, birds, flies, beetles, bats even. But pollination can also take place because of the wind. So if you've ever seen one of these dandelions that has all of its fuzzy little seeds along the outside as it starts to go through its life cycle, we know that the wind can pretty easily blow those seeds off the dandelion, carry them onto another flower or another part of the grass, and we'll get more dandelions that grow from those seeds. But humans can act as pollinators too. So maybe when there's not any wind, maybe you've picked a dandelion and blown the seeds across your yard. Or even if you're walking through a field and you disrupt the plants that are growing, you might help to carry some of that pollen on your clothing and distribute it around the ground. You could help to spread seeds or pollen that way as well. So we'll be dissecting a flower here, and some people might not consider this to be a dissection, but it really is. Flowers are living things that have certain structures that are able to help them go through their life cycle. So lots of different structures for lots of different purposes. And we'll be dissecting a daffodil today. Daffodils are great for this dissection because their parts and structures are really easy to identify. And for this, you don't need very many different tools. You only need a couple things. So on my tray, I've got some dissection tools. And even though my scissors look pretty awesome there, we'll go ahead and turn off our virtual background just so that it's not distracting. So for this dissection, you will need a pair of scissors that'll help you to cut the flower from outside if you are getting it from the wild. We'll use those a couple times. Tweezers are super helpful for this dissection. Tweezers will help us to get some of those smaller structures out of our flower as we start to identify them. And then I also have a scalpel. This is optional. You don't necessarily need the scalpel for this dissection. You could use a knife, but of course, if you're using a knife, you should have parental supervision. So this could be a great activity to do as a family this weekend, especially since it is going to be Mother's Day on Sunday. Maybe someone in your life would be willing to donate a flower to dissect as a group with your family, because this is a super fun activity. The knife is optional, and we'll talk about why we need it for this dissection, but if you don't or aren't comfortable using a scalpel or a knife, you don't need it, and we'll talk about why that is as we go through our program. So we said that we'll be dissecting a daffodil, and daffodils have really, really easy to identify anatomy. So I'm just going to trim the stem of my daffodil off the flower so it's a little bit smaller and easier to work with there. And that actually brings us to this chart as well. So what I've done before the program is I've just labeled this piece of paper with the different pieces of the flower that we'll be looking to identify as we move throughout the program. So I've written down the different flower parts, and we don't need to worry too much about those just yet because we'll talk about them together as we go. But we've already gotten to one of these items. So we've already trimmed the stem just a little bit, and so we can actually cut a piece of the stem and leave it right there on the paper where it's labeled as the stem. Does anyone know what is the purpose or the function of the stem? So the stem is the structure that we're identifying, and so we've identified the stem. Can anyone tell me what is the function of the stem? Why do flowers have stems? What do they do for the flower or the plant? Jason tells me one thing that the stem does is it helps to bring water to the flower. So the stem acts almost like a straw. And so it is actually able to draw up nutrients like water up the stem and carry it to the part of the plant on top, which we call the flower. So the stem is great for absorbing water. We said other nutrients as well. Perfect, everybody. Lots of great answers coming in. Now, what else can the stem do? If we didn't have a stem, would the flowers be off the ground? Probably not, right? So the stem also helps to hold up the flower so that it can be pointed towards the sun, so that it's up above the ground for pollinators to have access to. So the stem is our first structure that we've identified. Pretty noticeable. All flowers have some sort of stem. And this is a good time to talk about the differences between flowers. So we talked about that all flowers look different, that maybe they look different to attract different types of pollinators, or they look different because they exist in different environments, but they share common anatomy. Just like humans and sheep have very similar anatomy in their eyes, flowers have very similar anatomy between different species. So over here on the left, this is a lily. And if you do have access to a lily to dissect, I highly recommend them. It is very, very easy to see all these structures inside a lily, but we're dissecting a daffodil. And you can see that some of these words like anther and filament overlap between these two pictures. So even though these two species of flower are totally different, we're identifying common structures that have common purposes or functions. All right, let's take another look at our daffodil here. And we can pretty easily, without really doing any sort of dissection, start to identify some of these structures. The first thing that is pretty easily identifiable on a flower are the petals. And so you can just take your petals, you don't even need a tool for this, and you can just start to pluck some of them off. And we'll set that down on our guide where it says petal. We'll take off maybe 3 of our petals here. Take off this one. All right, so we've taken off 3 of our petals. And inside the petals, we have another structure. This right here is actually not a petal. It's a pretty unique structure. So just like certain animals will have certain adaptations that make them unique, this is actually called the corona. It's called that because in Latin, corona means crown, and so it almost does look like a crown, but it's also called the trumpet as well. And so that's another one of those structures that we can very easily take off of our flower. This time we'll use the scissors so that we can take just half of it off and then stick it down on our dissection guide. There we go. Just trim it here. Remember, always being very, very careful because we want to leave all of our other structures intact. And when we're using scissors, of course, we always just want to make sure we're being very, very careful not to hurt ourselves either. All right, so that is the corona, and that's actually so big that it's covering up some of my words. So maybe we'll just leave just one piece of our corona there on our sheet. So we've got petals and the corona, or what we might also call the trumpet. Now we also, on a flower, have what's called the sepal. The sepal is this green structure that meets the petals on the base. And at one point in this flower's life cycle, the sepal entirely covered the entire bud of this flower. The sepal's function is protection. So while this flower is developing all of its parts and still growing inside the bud, the sepal is really tough and it's there to protect the flower. And so we can, again, just use our finger for this one and almost just pluck our sepal off. If it's easier to use your scissors, you can do so, but I have pretty good luck just kind of peeling it carefully with my fingers and placing it down on our dissection guide, or our labeled guide there. All right, now the next thing on our labeled diagram is called the spathe. And the spathe is another one of those unique structures. Only certain species of flowers have what's called the spathe, and the spathe is this leaf-looking structure here. This was also there for protection. Once the flower has outgrown its bud stage, once it's outgrown that budding phase of its life cycle, the spathe starts to dry up. So the nutrients that once filled the spathe to help keep the flower protected, those were diverted into the flower and absorbed as nutrients. So since the spathe is no longer needed at this stage in the flower's life cycle, it's nice and dry, really, really easy to find. Again, it almost looks like a leaf, but we would call this the spathe. And so we'll just set that on our guide right there. Now this one is fun, especially since I plucked these flowers out of the ground, they are starting to drop their pollen. That's a natural reaction of flowers once they start to die, is that they want to release their pollen. They want to start pollinating other plants. That way their life hasn't gone to waste, because that's really one of the main goals of life, is to reproduce, is to have more progeny, especially in plants. So we can take the flower, and if we just hold it over our tray here, we might be able to see it better if we do it on this blue part. We just shake it. It's very, very fine. Let's see if we grab a different one, if we might be able to see it a little bit better. Shake our tulip there. But these fine flecks, you can see them just above my finger there and there. Those fine flecks, those are pollen. And pollen is helping to carry that genetic material to the next flower. So sometimes if you shake your flowers, you can get a good amount of it off. You can see a little bit all over my paper here, but we'll just try to shake off any that we can right onto our paper just by tapping it pretty gently over the paper here. So again, this dissection really doesn't take very many tools or expertise. It's very, very easy, and you can do it with just a few simple materials. Now that we've taken out the exterior parts of our flower, things like our petals and our sepal, we're going to take a look at the internal anatomy of the flower. This is where our reproductive parts of the flower come into play. So if we look at our diagram again, we have both male and female parts on flowers. So flowers will have both male and female parts. We'll start by first looking at the female parts of the flower, which collectively we can call the carpel. So the carpel includes the stigma, the style, the pollen tube, the ovary, which is this whole piece here, and the ovule. Now that's on our lily, where they're all pretty easy to identify, but we can find those same structures over here on the daffodil. So we have the stigma, which is the very tip, the very top of the female organs. We then have the style, which includes the pollen tube, which leads all the way down to the ovary. And the ovary contains those tiny little circles, which are called ovules. So the female part of the flower is able to trap pollen, carry it down the pollen tube in order to fertilize the seeds or the ovules inside, which can then turn into seeds or fruits or new flowers eventually. So let's take a look at those parts of our female flower here. So we can find the stigma and the style pretty easily. They are the tallest part of our daffodil. So right here, especially if I use my tweezers to point, we can see that we have the stigma, which is the very top of our female anatomy of a flower. We then have the style, which is the long tube that leads all the way down to this round part of the flower, which is super easy to see on a daffodil, which is called the ovary. Before we cut that in half, it'll be easier if we first remove the male parts of this flower, or some of the male parts of this flower, which include the anther and the filament. So the filament, just like the style, is kind of a structural piece. It's meant to hold up the anther. And the anther is where all of the pollen on this flower comes from. So the anther and the filament are the male parts of this flower. They carry the pollen, which is able to fertilize another flower or germinate another flower. So when we shake the plant, that pollen can fall off the anther, which is what we found on the tray. So let's go ahead and let's take off some of those anthers and filaments. So we can just very carefully take our tweezers, grab just one at a time, pluck them off at the very bottom. And you can again do this with tweezers, or even your fingers work just fine. You can see that this one had a little bit of spider webs inside it, which is kind of fun. So spiders might not be one of those pollinators we talked about, but they certainly would help to pollinate a flower if they did crawl inside, get themselves covered in pollen, and then go visit another flower to do the same thing. So this is one anther and one filament right here. So again, the anther is the top part, this brown piece, which would carry the pollen or produce the pollen. And then the filament is the structural piece that holds it up in order so pollinators can reach it more easily. So there we go. We'll split them apart. So if we take our tweezers and pick that off, scoot our diagram up just a little bit. So we've got filament, the stalk, and the anther over there. And let's take off a couple more so that we can more easily see our female parts of the flower. So we'll go ahead and we'll tweeze off one more maybe. So we've got another male part, which collectively we would call it the stamen, which I've written over on the side. So the stamen is both the anther and the filament. And so we'll just take that, pluck it off, anther over there, which is the pollen piece, and filament right there, which is the structural piece. So again, structure always relates to function, right? So our filaments help to hold up the anther so that pollinators are able to access them more easily. Now we can remove our female pieces more easily. So we said that this entire structure right here, this is a female structure. We just break it off at the top. We have the stigma, which is just this very, very tiny piece at the top. And the easiest way to remove that might just be by taking your pair of scissors and just cutting just the very top off. Now this daisy only has one stigma and style, but some flowers, some species of flower have multiple carpels or multiple stigmas and styles. Just like this flower had multiple anthers and filaments, some species of flowers can have multiple female parts as well. Now this is the part that's very, very cool. If we want to take an up-close look at the ovary, this is that step that would require something sharp like a knife. So I've got a scalpel here, which is incredibly sharp. So always use caution if you're working with something like a scalpel or a sharp knife, or always have an adult there to help you. That would be an even better idea. So all we're going to do is take our flower, turn it on its side, making sure to keep my fingers away from the scalpel, making sure you can see, and just cut right through the center. And we've now just split our ovary in half. So we can just take the top piece, split it open, which allows us to see those ovules inside. So Again, the ovary is the entire structure. It's this entire round piece right here. And the ovules are actually really easy to see in these daffodils. They're quite large in terms of ovules. We said that these ovules, once they are pollinated by pollen that travels down the stigma and style through the pollen tube, can then become new seeds or new flowers at a different part of their life cycle. So super easy. Again, if you are going to do that part of the dissection, be very, very careful. Always make sure you keep your sharps away where you're not going to accidentally touch them. And then we can just add our other half of our ovary. We'll trim it off so that we don't get anything extra. And we can just place our ovary onto our labeled diagram here in the spot that says ovary. All right. So now we've pretty much dissected our entire flower. I really enjoy looking at the piece that's left over. Getting this good cross section of a flower is really great for taking a look at all of those structures. They almost look exactly like our picture or our diagram that we found, right? So we can still see the corona or the trumpet, our petals, our sepals. That's the ovary from the outside. The inside we have our ovules. These again are our male pieces because we removed our only female piece of this flower. So we removed our only stigma and style, but we left behind 3 of these anthers and filaments. So pretty interesting dissection, very, very easy to do. And it, again, does not take very many tools in order to do this dissection. But now let's talk a little bit more about how these processes work. So we've identified the different structures of our flower and their function as well. But let's talk a little more about how these processes tie into plant life or plant reproduction. We saw that on this daffodil, the flower had both male and female parts. So it had the female parts here in the center and the male parts on the outside. Flowers that have male and female parts are able to self-pollinate. So first, let's just take a look at this yellow flower. Now, think about this process of self-pollination, which means that the pollen from the male parts of the flower travel down the pollen tube, they germinate or pollinate this flower on itself, and then will turn into more flowers. But let's think about this process. In this example of this yellow flower going through self-pollination, what do you think the new flowers will look like? Will they look like this flower? Will they look like this flower, like this one? Maybe some sort of combination? What do you think? What might this flower look like after this process of self-pollination? What will the new flowers look like that come from the ovules of this plant? Jason in Zoom tells me that he thinks that this flower or the new flowers produced from this process of self-pollination will likely look just like the parent organism. And that is a great thought process, right? Because if we think about where the genetic material comes from, right, that genetic material is only coming from this organism. It's not getting a mixture. So in humans, right, we get a mixture of genes from both our parents. We get some genes from the mother and some genes from the father. And we end up having a combination effect, right? Because humans are diploid. We get one copy of each chromosome from each parent organism. But in this case, all of the genes come from the same parent organism. So it is likely to look the same. But then in cross-pollination, which is really the goal of these species of plants, that's why they keep their parts so separate, is they really would like to cross-pollinate, meaning that some pollen will need to travel from one flower through a pollinator, whether that's a bird or the wind or a bat or a person, onto the stigma and style of another flower to create new flowers. These ones, the result of cross-pollination, right, likely won't look exactly like their parents. They could turn out to be a combination of these flowers. So if we mix red and blue, it's possible that we could end up with a purple flower, or maybe it could end up with a mixture of some pink and some blue flowers. because there's a mixture of genetic information coming into these new plants. That is an important part of nature because that is how selection happens. The most fit flowers, the ones that grow the best in their environment, whether that means they're the biggest or have the deepest roots or are the most beautiful to pollinators, those ones are the most likely to pass on their genes between different flowers. And so we'll likely end up having flowers that are very well suited to survive in their environment because those ones are most likely to pollinate other flowers. The very last thing we'll talk about is these two different types of plants. So there are two different types of flowering plants. We can call them either monocots or eudicots, and they're a little bit cut off. These are one word. I think it's just squished like that to fit in this diagram. But monocots and eudicots have different traits, and we can classify based on what the seed looks like, if we could take an up-close look at the root, if we could take an up-close cross-section look at the stem, we can base it on the leaf and on the flower. So take a moment, think about the different parts of this diagram. Think about the looks we've gotten at a daffodil, and here's a photo of a daffodil outside. So it's got all of its pieces intact. Tell me, based on this diagram, what kind of flower or plant is a daffodil? Is it a monocot or a eudicot? And this is part of the point of doing dissections. This is so we can take an up-close look, right? Because we may not be able to see all of these different structures if we hadn't cut open this flower and got a closer look at some of these structures. This would be even easier to do if you could do this dissection up close so that you can take a close look at some of these things that I might not be able to show you on camera very well. Things like the stem, what the cross-section of the stem looks like, or a cross-section of the root. Since I didn't pluck the roots of my flower, I didn't quite get that part for us to look at, but take a look and then tell me in the chat or the comment feature, is this a monocot or a eudicot? Great answers starting to come in. We've got some votes for both answers, which is okay, but let's go through. We really only can take a close look at a couple of these features, especially based on this photo. So I do have a leaf that we can take a look at up close as well. So our diagram says that the leaf of a monocot has veins that form in a parallel pattern, whereas a eudicot has leaves that form in a net pattern. So take a close look at those, and then let's cut our studio camera, or cut to our studio camera, and take a look at the leaf. If you take an up-close look, which our camera can show us, we can see that those lines run parallel. So that could be a good indication that this is a monocot, not a eudicot, because there's no netted pattern on this leaf. But just to double check, let's go to our other feature, which was the number of petals or flower parts and how many they come in. So it says that the flower parts, like the petals, will come in multiples of 3 on a monocot. So let's take a look. I've got a fresh daisy that we can take a look at, or a fresh, daffodil somewhere. This one's a little smaller, but same type of flower. And if we count the flowers, so remember that this is not a petal. This centerpiece, that's what we call the trumpet or the corona. And we took off some petals from our flower earlier, but we can pretty easily count the ones here on this fully intact flower. So if we don't count the trumpet or the corona, we have 1, 2, 3, 4, 5, 6, which is a multiple of 3. So that checks 2 of the boxes, telling us that this flower, a daffodil, is a monocot, not a eudicot. But you can dissect any flower that you find in your garden or are able to pluck, and maybe the one that you find is an example of a eudicot. So it would have petals in multiples of 4 or 5 instead of just 3. So this is about the end of our program here. If you have any questions about our dissection so far, feel free to type them in the chat. We can save a few minutes to talk about your questions that you might have. see. Yeah, lots of people talking about monocots and eudicots now. Cassie tells us that there are, in fact, some species of plants that have separate male and female flowers, and that is absolutely true. Most commonly, flowers will have both male and female parts, and they can actually be very interesting looking. So they can actually be a little bit tricky to identify. So if we take a look at something like our carnations, we'll take just one of those off. This is a little bit harder for us to see those different male and female parts because everything is so condensed inside. It might take you a really long time in order to count the petals on a carnation. So maybe if you were looking to classify it, you might want to look at some of those other features like the pattern on the leaves. So let's go ahead and let's try to cut this one open though, because it does still in fact have male and female parts inside. So if we cut it down the center, just like our daffodil, we can see, we remove some of that deep inside the flower, we still have our stigma and style attached to our ovary right there. Very, very tricky to see because they're a bit smaller and we dyed them with food coloring, which makes them kind of funny looking. But we still have an ovary attached to female parts, our stigma and style, and then this is an anther, very, very small, which is another male part of the flower. So most flower species, especially the ones that we have in our garden, have male and female parts, but it's true that some of them can produce separate male and female flowers. Jason he asked about dandelions, and it also has probably too many petals for you to count. So maybe you just take a look at one of those other features or factors of a monocot or eudicot. You can always look at the leaves instead, or you can take a cross section of the stem or the root and try to find these identifiable features as well. Let's see. So Jamie asked, why are there male and female parts? And that's because, again, structure is related to function. Because we need the male parts in order to produce the pollen, and the female parts contain the ovules, which will turn into new organisms. So male and female parts all have their different functions, but in different species of plants, we can have fused pieces or different pieces, but these are the common ones that are pretty easy to identify. Great questions, everybody. All right. I'll keep holding out to see if anyone has any last questions. But of course, at this time, we want to thank our sponsors like Ford and Denso, who allow us to bring these programs to you every weekday at 2:30 p.m. I hope you're enjoying the programs and that we'll see you back here tomorrow at 2:30 for another exciting STEM program with our Echo Distance Learning program. If you are enjoying these videos, feel free to share them on your Facebook page for others to enjoy. And you can also check them out on our YouTube channel as well. So with that, I hope everybody has a great rest of their day, and I hope I'll see you back here tomorrow for some more Echo Live.